A bioartificial liver device, BAL, is an artificial extracorporeal supportive device for an individual who is suffering from acute liver failure. History. Early history. The first bioartificial liver device was developed by Dr. Kenneth Matsumura and was named an Invention of the Year by Time magazine in 2001. Liver cells obtained from an animal were used instead of developing a piece of equipment for each function of the liver. The structure and function of the first device resembles that of today's BALs. Animal liver cells are suspended in a solution, and a patient's blood is processed by a semi-permeable membrane that allows toxins and blood proteins to pass, but restricts an immunological response. Development. Advancements in bioengineering techniques in the decade after Matsumura's work have led to improved membranes and hepatocyte attachment systems. As time has progressed, the sources of hepatocytes have increased. Cell sources now include primary porcine hepatocytes, primary human hepatocytes, human hepatoblastoma, C3a, immortalized human cell lines and stem cells. Use. The purpose of BAL-type devices, currently, is not to permanently replace liver functions, but to serve as a supportive device, either allowing the liver to regenerate properly upon acute liver failure, or to bridge the individual's liver functions until a transplant is possible. Function. BALs are essentially bioreactors with embedded hepatocytes, liver cells, that perform the functions of a normal liver. They process oxygenated blood plasma, which is separated from the other blood constituents. Several types of BALs are being developed, including hollow fiber systems and flat membrane sheet systems. Hollow fiber system. One type of BAL is similar to kidney dialysis systems that employ a hollow fiber cartridge. Hepatocytes are suspended in a gel solution, such as collagen, which is injected into a series of hollow fibers. In the case of collagen, this suspension is then gelled within the fibers, usually by a temperature change. The hepatocytes then contract the gel by their attachment to the collagen matrix, reducing the volume of the suspension and creating a flow space within the fibers. Nutrient media is circulated through the fibers to sustain the cells. During use, plasma is removed from the patient's blood. The patient's plasma is fed into the space surrounding the fibers. The fibers, which are composed of a semi-permeable membrane, facilitate transfer of toxins, nutrients and other chemicals between the blood and the suspended cells. The membrane also keeps immune bodies, such as immunoglobulins, from passing to the cells to prevent an immune system rejection. Comparison to liver dialysis. The advantages of using a BAL over other dialysis-type devices, e.g. liver dialysis, is that metabolic functions, such as lipid and plasma lipoprotein synthesis, regulation of carbohydrate homeostasis, production of serum albumin and clotting factors, etc., in addition to detoxification, can be replicated without the use of multiple devices. There are currently several BAL devices in clinical trials. A series of studies in 2004 showed that a BAL device reduced mortality by about half in acute liver failure cases. The studies, which covered 171 patients in the US and Europe, compared standard supportive care to the use of a bioreactor device using pig liver cells. See also: artificial extracorporeal liver support, liver dialysis, tissue engineering. References.